The overall goal of this procedure is to create a multi-layered cell culture scaffold. This video will demonstrate how to produce a 2D cell culture matrix incorporating the adhesion peptide RGDS in alternate layers to separate regions of C2-C12 cells. This is accomplished by first tethering RGDS peptide to an acryloyal macromer. The macromer peptide combination is then tagged with a Lexafluor dye to allow for visualization of the peptide-containing layers of the multi-layered gels. The second step is to form the molds by cutting them from a silicon sheet and sandwiching them between two glass slides. Next, after pre-mixing individual components of each layer, solutions of PEG diacrylate and PEG diacrylate with PEG RGDS Alexa 350 are alternately layered within the molds. Gels are polymerized by UV irradiation. The final step is to seed C2-C12 cells on 2D multi-layered gels in order to examine how matrix composition affects growth and attachment of the cells. Ultimately, bright field and fluorescence microscopy are used to visualize the selective growth of C2-C12 cells on the scaffolds.
The main advantage of this technique over existing methods is that it's a simple and a cost-effective method for creating multi-layered 2D and 3D cell culture matrices. It does not rely on need for expensive instrumentation. While the interfaces between layers are contiguous and structurally integral, there is no mixing between components of individual layers.
This method can help answer key questions such as how cells migrate and polarize in response to patterned biological, chemical,